Today we're going to be talking about nutrigenomics, metabolomics in the context of nutritional assessment. It's really important to be able to identify the imbalances. And as last week he discussed, you need to be able to identify dysfunction so that you can create function. Sorry, I'm getting some feedback on you, Carmen, so I'm going to mute. But from you guys, I'd just be curious. Feel free to turn cameras on, microphones on, if you feel comfortable. You can also use the chat here. But how many of you are currently seeing patients and doing some sort of nutritional assessment? This is Dr. Nacarelli. I met, I think in May, we crossed paths for the first time, but I was really impressed with Dr. Nacarelli for a couple of reasons. Number one, he is a PhD and a naturopathic doctor, which is pretty rare to find. You don't find a lot of people that have a good understanding of science and how that works, but also have a naturopathic doctorate. So I find that to be really interesting. But from that is clinical expertise is really guided by that. So he not only takes, like, you have a solution to everything, it seems like. I can't remember. We went to some, what was at the Persian restaurant? I was like, man, I overate. Panise ordered all these kebabs and stuff, and I'm sitting over here with a heavy stomach, and he's like, oh, I got this herbal thing that I did. You spray these bitters in the back of your throat, and then, it goes down and you digest better. And sure enough, that thing worked really, really well. Hopefully I don't overeat too often to where I need that on a regular basis, but just he always has some sort of trick up his sleeve from a naturopathic standpoint, and it's always vetted out from a scientific standpoint. So I really love that about him. But again, today we're going to be talking about clinical assessment of nutrition and doing that within the context of nutrigenomics, metabolomics, and some of the old tools that you're used to as well, like the questionnaires, the intake forms, etc. So I'm going to hand it over to Greg, and he's going to take it from here. Just remember, once we get to the Q&A, I would love it if you guys can turn microphones on at the very least.
Thank you so much for the introduction, Micah. And yes, that bitters tincture, it was wormwood, absinthium, and gentian, and I think some dandelion root in there. Super, super bitter. You had doubts at first, but after a couple sprays, you're like, woo!
Yeah, worked really well.
It was strong. We needed it. But okay, so I will go ahead and share my screen. So as Micah mentioned, we will be talking about nutritional assessments in clinical practice. This is a very large topic, and so in working with Theriome, I noticed a large spectrum in areas of expertise that various clinicians had. And there are some people that had not worked with nutrition in clinical practice. It was new to them. And so we wanted to have some type of foundational underlying for people that, okay, whether you're an expert in nutrition and that's all you've ever done or you've never done it ever, what's kind of a baseline of competency that people should have for nutrition? And nutrition is a very large topic, and especially when you're working with patients, you want to be able to assess nutritional status accuracy. So this includes, many different areas of it that I'll be covering today and then also next Friday as well. So again, this is not interpreted to be medical information, so please see your physician to accurately treat or diagnose any conditions. So Mike already did an introduction for myself. So again, I'm a naturopathic doctor and PhD, which is pretty rare. I know of like maybe 2 or 3 other ones, though I don't know of another naturopath who's a PhD in genetics. I think the other one is epidemiology and the other one is psychology. And yes, I believe in using omic information to help make assessments and diagnoses more precise. And I believe that by not doing imprecision medicine, which is just not using any omics information, we could have better adherence to protocols, better outcomes, and less side effect profiles, and just overall better healthcare experience. So a quick story. I used to be pretty overweight, and I didn't know I was overweight at the time. When I was in grad school for the first time, I was having fatigue issues. I was constantly bloated. I had inflammation issues. I couldn't think. And I didn't know it at the time, but I was probably like 215, which is well above a healthy weight for myself. And I saw a bunch of specialists I did a stress test. I did a CAT scan. I was thinking, what the heck's going on with my body? And it took me going to my primary care physician, which was this old, wily, veteran medical doctor. He had gray hair, just kind of told it like it was. And uh, I went to him and I'm like, I don't know what to do. And he kind of looked at me. He gave me a couple back taps to my gut, and he was like, "You probably should lose some weight. You should really focus on your diet." And I was like, "Whoa, this is the first time I ever heard somebody tell me that I needed to lose weight, one, and that I had to focus on my diet." Now, it was a very crude way to do it. He didn't give me any guidance on how to do this, but it was the first time that I became aware that this is something I needed to focus on. And so I would like to instill within you all a better way of having that conversation with the patient, giving them a blueprint for how to assess this, and be aware of the tools that you have at your disposal to assess this in order to help someone along. For me, I just started doing a ton of research on my own, and that helped me to then eventually, I wasn't a clinician at the time. This was before naturopathic school. But then through research, eventually I got myself healthier, and then I went to naturopathic school to help people with not just nutrition, but other areas of natural medicine, like I had been helped. So other than that story, why is it important to assess in clinical practice? So in general, poor diet is responsible for around 50 billion per year in US healthcare costs, which is astronomical. 45% of deaths from chronic diseases such as heart disease, stroke, type 2 diabetes, etc., are due to poor diet. And for one of the biggest killers, cardiovascular disease, 30% of all cardiovascular disease deaths are attributable to diet. So that's pretty substantial. When you look at the contributors to chronic disease, 30% of US adults are clinically obese. This isn't just overweight, this is obese. That's a very significant portion. And roughly 60% of all US adults have at least one chronic disease, and I believe 30% have at least two or more. So not good. We're spending a ton of money on healthcare, and we're having poor health outcomes. As you can see in this graph at the bottom right, just the incidence of disease, chronic disease of all different types, heart disease, obesity, cancer, diabetes, and the cost is just continuing to go up. So we need to focus on chronic disease, and we need to focus on diet in order to help manage this. So within this, so for part one today, we'll be talking about dietary assessments. We'll talk about different types of dietary assessments in terms of 24-hour diet recall, food frequency questionnaire, diet diary, and different app-based assessments, which are probably the most commonplace dietary assessment available. We'll talk about nutrigenomics, anthropometrics, and physical exams. And then next week, we'll get more into lab stuff. So we'll look at conventional labs, different types of samples, metabolite-based testing, and then microbiome-based. So for 24-hour diet recall, as the name suggests, it's pretty simple. Over the last 24 hours, what has that individual consumed? And usually I figure out what time of day it is for them, and then I will backtrack. Okay, if it's noon, did you have lunch? If not, did you have breakfast? Some people are doing intermittent fasting. Some people are skipping. So whatever their last meal was within the last 24 hours, get it from them. So whether it's breakfast, lunch, dinner, if they've had snacks, what fluids have they consumed? If it's not a typical day for them, ask them, okay, what in a typical day do you consume? This is easy to do. You could do it within the first visit or every visit, just to kind of get an adjustment, or if you get familiar with what their diet is.
Can I interject real quick?
Yeah.
For everybody that's interested in getting some handouts, I'm going to compile some handouts for you guys that's based on what he's talking about here in nutritional assessment. So you have something to walk away with. In addition, I can put some of the slides in here as well. And I'll send out the recording of this in case we come across something that's valuable for you. You want to be able to remember it, but you have to do one thing. You have to put yes just into the chat for me so that I can know I know who wants to get one or get the handouts, get the slide set and the recording. Sorry, continue on. But yes, put yes into the chat here.
Absolutely. Yeah, that's another thing too. So some of these you might want to send home with your patient for them to fill out, particularly for the next one, the food frequency questionnaire. So, and there's a couple of standardized ones that you have access to. So now the drawback for a 24-hour diet recall is that the sample size could be small. So within the last 24 hours, how typical of it is that? I'll tell you, like 90% of the time, every time I ask somebody what their 24-hour diet recall, it's like, oh, this is not usually what I eat, but it's usually includes some things that are unhealthy. So they're kind of, um, trying to account for that. The recall may be biased. They may selectively avoid certain foods if they feel like they're being judged. They may forget others. And then some people with memory issues, they just might not remember it all. So it's imperfect, but a quick way to assess nutritional status. There is the food frequency questionnaire. So this is where foods are listed and you find out how frequent are they consuming those foods in a given duration of time, whether it's less than once per week or multiple times a day. And so the advantages of doing this, it's inexpensive, it's easy to implement, it's more accurate than a 24-hour diet recall. But within this, you're lacking specific details about certain foods and brands. It can be time consuming for an individual to fill out. And how accurate it can be can fluctuate based on length of intake. And this could be paper, which might be an issue for some people. Some people don't want to fill out anything by paper anymore, which is understandable to a degree. But then there is the diet diary. This is when I first got started working with nutrition. I was using this heavily. One thing that's nice about this, you have them fill out what they're eating on a certain day, and you could have this for a week or 3 weeks or a month, however long you want them to fill it out. They could also list what their activities were during the day. And so if they had a particularly stressful day, you might see certain eating patterns. And, I've had people that say, like, for example, they're avoiding dairy, and I'll look at their diet diary, and then I'll just circle it for them all throughout the week where they were eating dairy. They're like, huh, how about that? I was eating dairy. It's like, yep. So, with this, there allows for more details, so that's beneficial. It allows the patient to be a little more mindful of what they're eating. Again, it's getting the patient to think about what are they eating, and then when they're writing down, because some diet diaries include how they're feeling during the day, so you could associate the foods with the feelings. This could be very informative for the clinician as well. This could also be triggering for them, though, to be writing down food. Some people don't want to do that, so there could be an issue there. Again, this is subjective. The patient is writing down what they're eating, so how accurate is it? And again, there's the issue with paper versus electronic tracking, which may be concerning for some. So overall, some of the issues with these, they're more generalized, they're not specific. They may be task intensive for patients just to be writing things down. It's subjective because it's the patient doing it, not you assessing what they're eating. It doesn't automatically calculate macronutrients, micronutrients. So it's worth considering if you don't want to get too intensive with it, and if you don't want to use technology. But I would say they are less common than technological options currently available, such as app-based nutritional assessments. So this has been studied. So they compared people that were using paper-based dietary assessments, like the ones I showed you, versus using a smartphone app to track their nutrient intake, their macros, and this was for individuals who were overweight. And basically, what they found was that they work just as well. And so if you use a dietary app and a paper app, they're both important for improving anthropometric measurements, that being weight and waist-to-hip ratio and things of that nature, which I'll get into later. However, if they work with a practitioner in using this, so they did a study where they had individuals using an app that calculated their calories, macronutrients, micronutrients. They did it themselves, and then they had them work with a practitioner to go over that information with them. And what they found was that when they worked with a practitioner, their weight loss improved, their body mass index improved, and overall the diet record completeness, this was pretty drastic, a 73.52% versus 28.91. So much more complete dietary record when they were doing this app tracking with a practitioner. So, these dietary apps work better if they're working with somebody. For using a dietary app, what are you tracking? You're looking at energy, kilocalories consumed, burned, how much you're over your calorie consumption targets, macronutrients, fats, carbs, proteins, micronutrients, all of them. Water, exercise, weight, some of them are integrated to track biometric information. In terms of apps, again, I'm not endorsing any of these, but it seems that MyFitnessPal is probably the most extensively used one on the market. though some people will say Chronometer is a little more accurate than MyFitnessPal. There's also a Noom Coach, which is sprung up in the last several years. It looks at energy intake and compares it to the daily energy recommendation. Samsung Health is supposed to be the most biometric compatible device on the market. And there's other ones, Lose It, Diet Plan 6. It seems like there's a bunch now. I would say, and some of them have more practitioner integration, like with MyFitnessPal. As a practitioner, you could kind of check in with people. So, you know, you want to have that in mind of your ability to access patients' health information.
It's helpful. Yeah, I use Chronometer just personally, but I'm not working with a practitioner on that.
Gotcha. So why would you use an app-based dietary tracking if you're being pretty stringent for your macronutrient, micronutrient weight loss goals? If you've had patients that are non-compliant with dietary recommendations, I did a rotation with a bariatrics a physician, and there was so many times where he would be having a conversation with a patient in office, and oftentimes it's patients that are super frustrated because they're not losing weight, and he's asking them, well, are you doing these different dietary things? And like, yes, I've been tracking my calories, I've been hitting my macronutrient marks. And then he would go into he was using MyFitnessPal, and he would see that what they were tracking was actually excess calorie consumption, snacking late at night. And he'd be like, look, I have the data right here. So it allows for more compliance with more targeted dietary recommendations, especially for people that are not having progress. Like usually what I'll do is I don't start off with tracking diet and macronutrients and micronutrients unless there is a strict or at least a lack of adherence of what dietary recommendations I'm making. I almost use it as a threat. Like, okay, if you're not going to follow my progress, I'm going to have you track everything to a T so we could follow that you are adhering to what is being recommended. But some people like that. Some people like tracking it because it makes them aware and they would otherwise forget. So this is kind of patient specific, and you have to feel out who is a good candidate for tracking using an app or not. Some patients don't want to share this information with you, so they might want to track it privately. There's something to consider. For people that have obsessive or disordered eating, this might not be good for them because this may become actually counterproductive to their health rather than productive. All right, let's talk about genetic impacts on nutrition. So this has gone by many different terms. It's been known as nutrigenetics, nutrigenomics, nutritional genomics. When I first started learning it, the way I learned it was that when you look at the impact of nutrients or nutrition on your genome, that's called nutrigenomics. And when you look at the impact of your gene on how you metabolize nutrients, it's called nutrigenetics. This is kind of not adhered to anymore. Pretty much nutrigenetics and nutrigenomics now essentially refer to the same thing. It's basically the interaction of your genes and genome with nutrition. By looking at this, you could get information regarding nutrition, or excuse me, nutrient insufficiency or excess, inclination or disinclination towards or away certain diets. You can extrapolate diet and nutrient associations with health and disease states. And then you could give information regarding dietary allergies and intolerances, amongst other key pieces of information. So some upfront genetics terminology. There is SNPs. When people discuss nutrigenetics, they're talking about this SNP and that SNP. There are many different types of variations that can occur in the genome. In some circumstances, there are deletions that are reported in nutrigenetics, which is basically have a nucleotide ATCG as our DNA backbone. One of those nucleotides is deleted, so that alters the expression of the protein. There's also copy number variants. This is found in like salivary amylase where people make more of this enzyme to break down carbohydrates. So copy number variant is another thing. But most of the time when we're talking about this, we're looking at SNPs. So this is within the genome at one single location, one of the nucleotides is different, and that is affecting the protein, which then affects what the protein does, and that's how the nutrigenetic association occurs. Now, what you'll also see is some entities use collections of SNPs to calculate something called a polygenic risk score. And so within this, the amount of risks that somebody has is added up. And so, for example, with this bell curve, you see at the bottom of the bell curve, these individuals have inherited less than 2.5% of risk variance. And then at the top of this bell curve, they have a high genetic risk, greater than 97.5%. In this example, this is looking at hypertension. But so for the people with the lower polygenic risk score, you see a later onset of hypertension and a decreased incidence of hypertension. Whereas with the higher polygenic risk score, you see an earlier onset and then a higher incidence of hypertension. So you could use this not just for hypertension, you could use this for basically anything. As you'll see, I'll use an example of cardiovascular disease. So that's a highly polygenic risk condition where cardiovascular disease isn't just due to one gene, it's due to many different genes. There's something called the omnigenic theory, which essentially posits, if you look at the genome, all of our genes have some degree of impact on health risks. It's just a matter of how much each gene is impacting it. And so the only way you would know is if you have some sort of calculation to figure out the degree of impact. And so, yes, maybe somebody has the ApoE4 risk gene or risk allele, and so they have an increased risk of either cardiovascular disease or Alzheimer's. But what about all the rest of their genetics? And so it's possible they might have an overall decreased risk rather than just looking at one single variant. And so that's why it's important to look at the entire genome versus just one individual variant, or even a couple handful of variants. Which is my issue with some nutrigenomics offerings on the market is some of them just look at a couple and then they make health extrapolations based on that, and it's largely inaccurate. And another part of this that I will add later is how do you assess for the outputs of this? So if somebody has a genetic risk for a nutrition deficiency or a disease, how do you assess that that is actually manifesting? Because the genetic risk is just a possibility. It's not actuality. So, question for the audience, and I would love to see if somebody could answer this correctly. So, what was the first nutrigenetic disease discovered? This was discovered in 1934 by a Norwegian scientist, and this was in babies and infants. They would be born with, or eventually would go on to have mental retardation and growth deficits. And what he found was that he analyzed their urine and he found that this elevated metabolite was present because there was an enzyme defect that was blocking their metabolizing of a certain amino acid. So I'll give the chat a little time to figure this out. Don't look it up. It's common. The last hint I'll give is you'll see it on some types of artificial sweetened bubblegum. It'll say, caution, do not consume if you have this condition.
I'm at a
5,
loss.
4, 3.
Anybody know? I don't know that one.
PKU, phenylketonuria. So basically, this is where individuals have a deficiency in this enzyme, phenylalanine hydroxylase. And so this enzyme is responsible for converting phenylalanine to tyrosine in individuals with this condition. And it's an autosomal recessive condition. So essentially, if your parents don't have it, they would have to be carriers for it. And so there'd be a 25% chance of you getting the condition. And so, and this is the enzyme that he found in the urine was this phenylpyruvate. And what's happening is instead of phenylalanine going down this pathway to make tyrosine, it's being converted into this metabolite as is measured in the urine. Tyrosine is a precursor for many different things, catecholamines, which give us energy. And so these people will also have lack of energy, which is not good. And so, as I mentioned, the children with untreated PKU, they'll have impaired brain development, intellectual disability, behavioral problems, epilepsy, and then lots of other complex health issues later on down in their life. The treatment of this, of course, is decreasing phenylalanine in their diet and consuming more low phenylalanine foods. And in some cases, you could even supplement directly with tyrosine to compensate for this nutritional deficiency. But so this is the first time that scientists found that there was a nutrigenetic association where there was a nutrient that our genetics predispose us to being impaired to metabolize. So why use nutrigenomics in practice? By doing this, we could help to personalize nutrition plans and avoid generic dietary plans. They surveyed patients and asked them if we were to base our recommendations on your genetics, would you be more or less likely to follow them? And 75% of patients said they would be more likely to adhere to treatment recommendations if it was based on their genetics. So by looking at their genetics in regards to nutrition, they'll have a better understanding of how genetics is impacting their health. If they don't already have a disease, by looking at genetic predispositions, it could lead to prevention of diseases, especially if you're targeting nutrition deficiencies. And then it allows more supplement strategies in terms of either supplementing with things you're possibly deficient in. Again, you could validate this with laboratory data and symptoms and physical exams, or you could avoid unnecessary supplementation. Maybe you don't need certain supplements because you don't have as much of a deficiency as you may have otherwise thought. So just a couple examples here. We'll talk about vitamin D first. So vitamin D, as most should be aware, it is made from cholesterol, and from exposure to sunlight increases this enzyme, which eventually upregulates the production of this cholecalciferol, this vitamin D precursor, which goes to the liver, then goes to the kidneys, and eventually becomes activated. And there's various enzymes in this pathway. And within these enzymes, there could be those single nucleotide polymorphisms, or SNPs, that alter the production of these active vitamin D metabolites. Why is this important? So these variants in these vitamin D SNPs are associated with decreased vitamin D levels, of course, but also altered immune function, osteoporosis risk, atherosclerosis risk, diabetes risk, cancer risk, and many other health conditions. So the impacts are far-reaching, especially knowing how important vitamin D is. Couple SNPs in particular. So whenever you see a SNP, there's always this thing, this RS before it, and then these numbers, which is essentially like a address. So this is a particular region in the genome is what this is referring to. And usually you'll see this in reference to a gene location. So GC is a vitamin D binding protein. So this SNP has been associated with the ability to make this vitamin D binding protein, which increases the risk of vitamin D deficiency. There's this intracellular receptor, RXRA. This SNP associated with response to vitamin D supplementation. Some people have a better response to that. And then there's a vitamin D receptor, which is very interesting. I was reading an article yesterday on just how even if you don't have a genetic risk for vitamin D receptor, viruses and pathogens will shut down your production of this vitamin D receptor. So you may have normal vitamin D levels, but if you don't have the receptor for vitamin D to bind to, you're not getting the actual benefits. So that's one thing to consider. But some people have a genetic predisposition to already have the dysfunctional vitamin D receptor, which has been found with many health conditions, one of which being increased obesity risk. Another one, TCF7L2. This gene is sort of infamous for the hunter-gatherer variant. And so there's one SNP in this gene where if you have the T allele, again, ATCG are our main nucleotide. So at this single position, if you have the T allele, you're more likely to have a poor response to grains and fibers and an increased risk of diabetes. Whereas if you have the C allele at this position, you're more likely to have a better response to grains and fiber and a decreased risk of diabetes. Ultimately, so this TCF7L2 is a transcription factor, is associated with a glucagon-like peptide, which you may be familiar as the Ozempic and Trulicity and Mounjaro, all those fun things. But it affects insulin production, glucagon, appetite, and food intake. And for people with this risk genotype, you will see reduced insulin secretion by beta cells, which will affect gluconeogenesis, which is sugar creation in the liver, whether your genetic association is weak or even strong, and then eventually you'll have an increased type 2 diabetes risk. Individuals with risk variants in this gene, they respond better to a Mediterranean diet and a low saturated fat diet. And then if you have one of these risk alleles, you may want to decrease your response, decrease your grain consumption based on whether you have the T allele or C allele. So we could focus on individual nutrients, we could focus on individual genes and how they're impacting things, and then we could focus on whole diseases. So for example, cardiovascular disease. This is a good example where you don't want to look at just one gene. You want to look at a collection of genes. So there are many different genetic variants that are associated with cardiovascular disease. There are many that they're just these random genes, just these random proteins or receptors that we're not sure, there's not a direct nutritional correlate. or an overt correlate that would make sense with cardiovascular disease. And then there's some that are very much make sense, right? So ApoE is involved in cholesterol transport. If you have risk variance in that gene, and you would obviously have increased cardiovascular risk. FADS is fatty acid desaturase, so it helps basically help metabolize omega-3s and omega-6s. That's malfunctionally of increased risk. Clock variance is our circadian rhythm. This is affected by protein intake, MTHFR, B9 metabolism, vitamin D receptor I just mentioned. CYP1A2 is involved with caffeine metabolism. So on and on. You could look at all these different genes, and if you just looked at one, you wouldn't have the full picture. And this is actually quite small. I mean, you're looking at probably over 1 million variants that have been associated with cardiovascular disease, but these are the main nutrigenomic variants that are associated with cardiovascular disease. And by knowing these, you may alter things such as your saturated fat intake. Whoops, what happened? Oh, am I still sharing?
My bad. I'm playing around with the settings over here, trying to make things a little bit better.
I thought I moved my
and
cursor too aggressively.
All right. You
Am I
should be
still
all
sharing
good now? Can you share again now?
Yeah, sure.
Sorry.
That's okay.
I just had to throw you off your rhythm. You were getting in too much of the rhythm there. So
no, that's all right. So yeah, we may alter things such as our saturated fat intake, whether we're should or should not supplement with omega-3 fish oils, our protein intake, our sources of protein intake, our polyphenol intake in our diet, our vitamin D amount, and different other dietary components. And again, we can support this information with nutritional intake information, looking at lab values associated with these markers. But our understanding the nutrigenetics can help us know our tendencies for some of these things. If you don't already have cardiovascular disease, at least with the goal in mind of preventing. So these are just a few examples. If you guys like this nutrigenomic stuff, we'll see if we could do more talks about this in the future. But there's a lot. There's a lot of different nutrigenetic associations from just looking at the nutrients alone, not alone how they are impacting our health and disease risks. So for example, overeating, the FTO gene is associated with that bitter taste. So these taste receptors, TAS2R38 and 16, associated with bitter taste. Sugar cravings, things like these SLCA2 receptors associated with glucose transport, LEP gene associated with leptin, which will affect sugar cravings. For fat, CD36 and FGF21 is how well fats taste for people. Some people don't taste fats that well, so they eat it more, and they have that high predisposition. Cilantro, of course, some people hate it. I have this gene. I cannot get anywhere near cilantro. It's OR6A2. You can look at genes that will affect people's response to certain macronutrients. So for protein, FTO, clock, TFAP2B. For fats, these fatty acid desaturase genes, ADIPOQ, which is adiponectin. Carbohydrates, into TCF7L2, like I mentioned. SI, sucrose isomaltase. This, for people, they'll have impairments in breaking down sucrose, so these people are more likely to be sensitive to FODMAPs. Amylase, their ability to break down amylose. Vitamins and minerals, there's basically genetic variations for all of them. Vitamin D I already briefly covered. B12, there's B12 transporters, TCN1, TCN2, FUT2, which is the secretor, non-secretor genotype. B9, there's a bunch. MTHFR is one of the most famous ones. B2 is a cofactor for the MTHFR gene, so some people need more of that. Vitamin A, BCMO1, some people do not convert beta carotene to retinol properly, so these people need increased dosing of vitamin A. Zinc, there's variations in the zinc transporters that are associated with decreased zinc levels and also increased risk of diabetes. Copper, superoxide dismutase. Copper is a cofactor for this, so some people need increased amounts of copper for that. Iron, there's a bunch. Transferrin, TPRSS6 associated with iron levels. Vitamin C, anytime you see SLC, think transporter. So SLC23A1 is a vitamin C transporter. And then for individual nutrients, there are some genes where some people have increased need there's some risk variance in genes where some people have an increased need for certain nutrients like CoQ10, this NQO1, which is an upregulator for all these antioxidant response elements, detox pathways. So there's some risk variance there. Lycopene, this is a really interesting one. I found out that I had a genetic predisposition towards lycopene, and I started supplementing with it, and I felt better since I started doing that. So go figure. And this is one of my favorite ones, berberine. So there are risk variants in PCSK9 where some people, you may be aware, there's PCSK9 inhibitors that people will use in order to bring down their cholesterol levels. And there's also LDL receptor risk variants. So some people, the LDL does not dock to the receptor properly to be brought into the cell. And they did a study where people with certain risk variants in these genes, they've responded better to berberine. So I'll actually look at this to see who is a better candidate for berberine or not, based on this and also their cholesterol levels. So applying this in practice, really to execute using nutrigenomics properly, you want to assess their dietary intake, health history, their health goals, and then you want to run a genetic panel then to assess for nutrigenetic associations. You want to make sure this is within your scope of practice. So, you're not diagnosing somebody with something with nutrigenetics. Like the PKU thing is more for metabolic disorders for pediatric doctors. If you are that, excellent, you could do that. You just don't want to get into the realm where you're diagnosing and treating people and it's not within your scope of practice. So make sure that is applicable. If you're doing this, again, I suggest looking at polygenic risk scores, not just individual variants, because you may not be getting the full picture. And even then, you don't just want to look at genetic risk. You want to combine it with other pieces of information like physical exam information, their health history and symptoms, and then laboratory data to confirm their nutritional status. I'm realizing why I already split this up, right? Because I think we're already at 45 minutes. So, and I have quite a bit more slides left. But so let's cover the physical exam piece. So if you're a practitioner and you're seeing patients, a lot of these things, you don't even have to see them in person. You could. There's ways that they could measure these things themselves at home. But you want to be able to capture this information and track it, combining this with your other pieces of information. This way, you're getting a whole picture of their nutritional health. When it comes to physical exams, there's anthropometrics, BMI, waist to hip ratio, bioimpedance analysis, which I'll talk about. Blood pressure has very direct nutritional correlations, and then various other areas that you can assess: their hair, their ability to smell, a bunch of things in the mouth, taste, their tongue, their teeth, their gums, skin, nail, sensation. So all these things you want to consider in nutrition clinical practice. So for the waist to hip ratio, it does vary based on whether you're a man or a woman. Generally, if you're a woman, you want to be 0.8 or below. Again, the waist to hip ratio is just below the 10th rib. You're going to measure it with one of those tape measures. And then the hip is where your greater trochanter is, where it's like that hard bony part right on just below your hip. And so for women, you want to be 0.8 or below. If you are above 0.85, your waist to hip ratio is elevated, which is associated with cardiometabolic risk. And then for a man, you want to be 0.95 or below. And then if you are a 1 or higher, that's associated with cardiometabolic risk. Bioimpedance analysis. I don't know why this, I'm sad that this was the best picture that I could find. This guy does not look happy. For bioimpedance analysis is basically they're connecting electrodes through certain parts of the body, and an electrical signal is basically sent from one electrode to the other. And so as that electrical signal is going through the body, it's measuring, is it hitting water, is it hitting fat, and is it hitting muscle? And as it does that, you can calculate these things in terms of the person's hydration, their fat-free mass, their skeletal muscle mass, their fat mass. There are many different devices that are available for this now. You don't have to go to some center to do this. There's scales that do it. You stand on this like metal plate and then you hold this metal bar. It'll send that signal through you. What I'll say is I don't know how accurate they are between each other, but if you are measuring this, just use the same device. This way you account for differences in the devices. But it is a useful way to track these things. As you're improving your dietary health and your nutritional health. And again, you have your patient do this on occasion and share this information with you, and you could see how they're improving. For blood pressure, I won't belabor this too long, but generally speaking, you don't want to be higher than 140. That's associated with stage 2 hypertension. I mean, generally you want to be below 120 to 129. I'm not a fan of hyper suppressing blood pressure. A lot of these people, then they don't get blood to their brain. So you want to have sufficient blood pressure, not too low, not too high. Generally speaking, when you're looking at blood pressure, nutrition associations are either sodium excess or deficiency. Potassium deficiency can increase blood pressure. Antioxidant deficiency, so our blood vessels need to be able to relax and contract. If there's a high level of oxidation status, the blood vessels are going to be stiff. Protein deficiency, carbohydrate excess, omega-3 deficiency going to affect our blood pressure, our blood vessels' ability to dilate and contract. And then many other vitamins and minerals, vitamin D, C, E, and our B vitamins associated with blood pressure. When we're looking at the hair, there's many different hair conditions that are associated with nutrient deficiencies. There's alopecia areata, which I'll just go briefly here, which is the image on the left. You'll see these kind of patches of hair that are missing, associated with protein, iron, zinc, copper, and biotin deficiencies. There's androgenic alopecia, which is this more characteristic, like widow's peak, if you will, on the top of the head and on the sides of the forehead. Associated with iron, zinc, vitamin A, vitamin D, vitamin C, biotin, and folic acid, and DHT excess and mitochondrial dysfunction. Dry hair, anything dry, you have to think fatty acids. So either omega-6s or omega-3s. For dandruff, you're also looking at possible zinc deficiency as well. Premature graying, that's that SOD, superoxide dismutase variation, where people just have excess oxide radicals, which leads to this graying. That's another gene where you could have a deficiency in SOD. And for those people, you want to especially make sure they have enough copper and zinc, because those are cofactors for that enzyme. So whenever I see premature graying, I think copper, zinc deficiency, or that superoxide dismutase enzyme. Corkscrew hair looks like this on the right. I've never seen that in practice, but apparently that is associated with vitamin C deficiency. And then hair thinning could be many things, but protein, zinc, selenium could be deficient in those individuals. For smell, so hyposmia, anosmia, nutritional deficiencies that are associated with this, it could be many different things: zinc, copper, iron, iodine. When you see something like this, an overall nutritional assessment, especially with labs, might be applicable because you don't know which one it is. It could be a whole slew of different things that are being impacted with this. And that's another thing too. You look at the pandemic, a lot of people lost their sense of smell, and it makes me wonder how many of these people, they're just nutritionally deficient, and then they're more likely to lose their sense of smell when exposed to something like COVID, which just disrupts that whole olfactory system. When you are doing a smell test, you have smell cards that you could use, kind of peel them off and let them smell them. Some scents that are applicable for this is coffee, lemon, cloves, cinnamon, smoke. I'll tell you a story. When I was doing this in clinical practice, when I was in school, the doctor said, "Oh, go get some coffee grounds and have them smell to do a smell test." And I'm like, "Where am I going to get coffee grounds?" They said, "Well, just go to the break room and just get some of the coffee from the pot." So I went to the break room, and without looking, I was just in a rush. So I open up the lid and I go take a big smell, and I'm like, I don't really smell the coffee. And I look, and there was coffee in there, but there was just a ton of mold. So I got a nice big waft of mold exposure, which was not good. So definitely make sure it's fresh and not previously used coffee, so you're not breathing in mold or whatever else is growing on it. Taste. So taste is another one. Can be many nutritional deficiencies associated with that. Vitamin A, all your B vitamins, E, copper, iodine, zinc. There's the zinc taste test. So for individuals, you could give them 50 to 80 milligrams of zinc, have them taste it in their mouth. If they taste it and they have an immediate unpleasant adverse taste, they're likely zinc sufficient. If they taste, they put the zinc in their mouth and they don't really taste it, or it tastes like water, in theory, they're very deficient. So being able to taste zinc could be indicative of impaired nutritional deficiency of zinc and impaired taste. One thing to note, so low zinc is very much associated with diabetes, and 70% of diabetics have some degree of smell and taste dysfunction. So especially things zinc deficiency in your diabetic patients. The tongue, there is absolutely a lot here. So there's various different things the tongue can look like. It could be red. You could have glossitis, which is inflammation of the tongue. You could have this kind of geographic pattern. You could have this rhomboid glossitis where there's this kind of patch in the middle here and the periphery is sensitive. They're each associated with different things. So if it's not true glossitis, it's just red and beefy, think niacin deficiency. For a highly inflamed tongue, it could be many different things. We mostly associated with B12, but it could also be protein and other B vitamins as well. Rhomboid glossitis, this one, think iron, folate, B12 deficiency. Geographic tongue could be iron, zinc, niacin, B6, folate. If their mouth is burning, that could be iron deficiency, vitamin C deficiency, niacin, B6, or folate. You could also look at tongue coatings. So in general, if you see a tongue coating, it could be iron, selenium, zinc, or vitamins, or basically your fat-soluble vitamins A and E, or C deficiency. With a white tongue, you have to think candida, could be some level of dysbiosis or thrush. With a yellow tongue, it could be hypochlorhydria or low stomach acid and dysbiosis. And then with a black tongue, never seen this, but it could be a smoker or sulfur dysbiosis. So lots of different things to look at with the tongue. There's also tongue fissures as well that are associated with different things. When you see a tongue fissure, it's also, you have to think of some level of gut inflammation or gut leakiness and nutritional deficiencies. With the gums and the teeth, so for gingivitis, which is inflammation of the gums, deficiencies of A, C, and D, periodontitis, which is right here, you can see this kind of degradation of your gums. Deficiencies of A, D, C, E, and B12 are big ones. Red gum line color, vitamin C, CoQ10, big ones. Easy bruising, so vitamin K, obviously, but vitamin C as well. Tooth decay, there could be deficiencies of minerals we're looking at, so calcium, phosphorus, fluoride to an extent, but that's kind of debatable, and then vitamins A and D. Attrition of the gums, vitamin D and calcium, and then hypoplasia, it could be vitamin A, C, or D. So lots of different vitamin deficiencies associated with just looking at the gums. For the skin, again, when you have anything dry, think fatty acids, but also zinc and A, C, and B vitamins could be associated with that. Follicular hypokeratosis, which is this one. It's kind of like the bumps on the back of the arm. I do think food allergies, when I see that, and food sensitivities, especially gluten, it can be also associated with fatty acid deficiencies, zinc, vitamin A, and B vitamin deficiency. Seborrheic dermatitis, essential fatty acids, especially cradle cap, zinc deficiency, vitamin A, riboflavin, big ones. Eczema, fatty acids, but also zinc, vitamin A, D, and E, just fat-soluble vitamins. Acanthosis nigricans, which can be occurring from excess glucose, cortisol imbalance. Chromium plays a particular role in this as well, but it's where you get these kind of hyperpigmentation in the skin folds as well. So something to think about, blood sugar dysregulation when you see anything like this. Again, going on skin, so ecchymosis, which is essentially this bruising here, you have to think any coagulation vitamins, so vitamin K, and then collagen-associated vitamins, so vitamin C, hyperpigmentation, protein, niacin, folate, and B12. Hair loss, protein, zinc, biotin, or excessive vitamin A can actually lead to hair loss. Acne, many different vitamins associated with this. EFAs, zinc, selenium, vitamin A, E, and some B vitamins. Melanoma can be associated with excess glucose, niacin, or chromium deficiency. And then poor wound healing, basically it could be a whole slew of vitamins, but especially protein deficiency, I would also consider, and zinc and vitamin A. The nails, lot that one could look at here. Couple that I'll highlight. So the absence of these moons, you should see these moons in your base of your nails. If theirs are missing, you have to think B12 deficiency. Pale nails, you can think iron. These vertical or horizontal ridges or lines, these bow lines, could be protein, fatty acid deficiency, iron deficiency, or B vitamin deficiency. Brittle nails could be basically any nutritional deficiency. Spoon nails as well. If you see someone with spoons in their nails, there's overt nutritional deficiency that's occurring. So that's another thing to consider. Leukonychia striata, so these kind of horizontal lines. Severe protein or zinc deficiency. So I would look at that as well. Sensation. So one key thing about sensation, If it's bilateral, it's probably nutrition associated. So it could be B12, folate, niacin, copper, thiamine, and B6, either insufficiency or excess. Because if you have excess B6, it's almost the same issues that arise if you're B6 deficient. So if it's bilateral, it's likely vitamin deficient. If it's asymmetric, it's probably not. It's probably non-nutritional. And with this, you could use a monofilament, which is a little stick with this little pointy thing, hit different areas of the foot, and let the patient know when you are touching it, and you see how accurate the patient is to sense that you are touching their foot. So I know that was a lot. There's a lot of content there. Mike, I believe this will hopefully be available for people to review if they want to relook at it. But overall, the take home here, make sure you perform a head to toe nutrition focused physical exam on new patients just to get an assessment of these areas of health that you could just see. You could look at a lot of these things just through a camera, right? You could have them, you could look at their teeth, have them stick their tongue out, look at their tongue, look at their hair. So you could do this wherever or however you want to do it. Track these changes over time. If you uncover nutritional deficiencies and as you help to make them nutritionally sufficient, you should see these physical exam findings change. And then if you have these suspected deficiencies, especially for these issues where there's a lot of nutrients that could be deficient, run labs to see, okay, how's their blood looking, how's their urine looking, depending on which metabolites you're looking at. But you want to confirm this with some labs.
Yeah, and just to fill in real quick too, this is a two-part series. We weren't initially planning on it being a two-part series. So next Friday will be a continuation of this where we get deeper into the metabolomics portion. And will you be covering nutrigenomics as well, or just kind of sticking to the? Give me a teaser for next week.
Yeah, I might talk about like what types of labs to run based on nutrigenomic things that are uncovered. So there will be some touching about that, but mainly sticking with the labs, metabolomics, regular labs. It's I'll be covering that whole area.
Okay, very good. So we're planning on doing this every week. I'm sure you guys have seen some of the emails. Obviously, the ones here have noticed. I've been sending out a couple emails a week just to announce whatever the Friday topic is going to be. I'd be curious to gain some feedback from you guys. So if I were to, and you can put yes in the chat or no, if we were to continue with Greg and we just go really deep with Greg and understanding how he thinks about these things versus, and we'll do that too, but would you also be interested if we brought in, say, an expert? So one of the guys that's using our test quite a bit, he's one of the top breast implant illness doctors in the country, just by volume of patients that he has. Would you be interested in hearing on different experts and how they clinically apply labs with what they're doing? Another example would be a doctor out of Texas who works with a lot of the professional golfers, and he's using our metabolomics panel for that. Would that type of stuff be interesting to you, or do you prefer to stick with one person and go really deep? So if you're interested in doing a one-on-one with Greg, he does one-on-ones and you're able to schedule this where you can sign up, talk with Greg. How long is it, Greg?
The meetings with me one-on-one, or half an hour, I believe.
30 minutes. So you get 30 minutes, to discuss with him. It is going to be geared around metabolomics because that's what we primarily focus on as a company. We are going to be launching our microbiomics. We've already done an early access launch to some select clinicians as we're gaining some more clinical insights and working with them for the next until we're anticipating January, February. At that point, we'll have another person that does the microbiomic side, or maybe Greg does some of that too. But anyway, if you want to talk one-on-one with Greg, go to that link. I'm also going to put it here into the chat. So click that link. You'll be able to sign up, talk with Greg a little bit more in depth, a little bit more about your clinic, the current labs that you're running and what you would like to be able to see out of your labs and if we have a match for that or not. Our goal is to basically equip the physicians by giving new levels of insight. So if you're not familiar with our test, we're doing a really wide breadth, a really wide profiling of the metabolism. We're actually covering 68%, which if you were to combine 6 different standard metabolic panels, including the oat test, you would get about a 19% coverage. So we're doing really wide breadth, and that does have a lot of applicability in various areas. So our goal is to equip doctors. But at any rate, what we're going to do now is we're just going to go into Q&A. I know we're running to the top of the hour here, which is the total time we had planned. But Greg, are you able to stick around past the hour?
Yeah, we do have that next meeting coming up, but yeah, we could hang
Okay.
out.
So we got about 20 minutes here available on our end. Says the link is not working. Anyway, I'll put it in again and double check to make sure it's working. But we have about 20 minutes here where we can do Q&A. So I'd love it if you guys turn on your microphones. That's the #1 preference. But secondly, if you don't feel comfortable turning on your microphone, just go ahead and put your questions in the chat. So please ask questions.
Well, my questions are going to be related to things I had earlier put in the chat, like as far as, I guess, how detailed or whatever is the testing going to be as far as the data being collated so that we can make a recommendation to the patient as far as what type of meal plan or meal program that they should follow, obviously based on the testing.
Yeah, so I think that depends on the program that you're using to analyze genetics. So one, you could look at individual SNPs that are looking at nutritional deficiencies. And again, I wouldn't just come up with a recommendation based upon that. I would look at other pieces of information, coupling that with labs in order to make assessments. There is options available for people to upload their genetics, and it comes up with a dietary plan. I feel like it's hit or miss. Sometimes it is accurate. Some dietary plans say, "Oh, you're a better responder to a ketogenic diet," so people increase their fat consumption, and then they wind up gaining weight, and they're like, "What the hell happened? I thought I was supposed to do better on fat," and then sometimes you get the reverse. So, those options exist. I think they are getting better, but I think a more multifaceted approach that couples all the information in terms of the health history and genetic predispositions and lab marker output would be the ideal piece of information in order to come up with a dietary plan. At this stage, I would say maybe, thankfully or not thankfully, the practitioner is still heavily involved in this process. It still involves a human being to look at each piece of information and make the determination. Though it would not surprise me within the next maybe 5 to 10 years, you could have all that information being assessed by AI and then coming up with a recommendation based upon that. But for now, it is still up to the practitioner's discretion.
Well, to me, that's kind of the point of the testing, and that's what I thought it was, that you'd be able to make a recommendation to the patient on how they should eat and what's going to be the most effective meal plan for them to follow. Like what we do is we're mainly see people that want to lose weight. Once in a while we'll have somebody who's who's a gym rat and they're looking for optimum nutrition. That's one of them, blue moon, not that many. But generally I deal with people that are overweight / obese. Some of them are morbidly obese. And of course they're coming to see us because, they want to get a solid recommendation on what's going to be the most effective thing for them. So anyways, I am not a doctor. I'm not a dietitian. We primarily, the product that we use mainly for weight loss is semaglutide tirzepatide, which is very effective. But my interest in your company is being able to have another tool at our disposal and something that we can recommend that's going to be beneficial to the patient in order to optimize their results, not only just losing weight, but to get these people as healthy as they possibly can be. So what would your suggestion be? I should look for what in the world of the medical field? Is this something a GP is going to be able to look over this test and understand all the details and what it's telling them, or a dietitian, or you need somebody that's more in a specialty?
Yeah. What you're asking for is, it's called personalized or precision nutrition. And the idea is that you get more precise by incorporating pieces of omic information, be it genetic or metabolomic or microbiomic. It is not absolute. And so you have to look at multiple facets of information. I can't emphasize that enough. So like, because otherwise, why would somebody even need to see a practitioner, right? Like, look at the study with the dietary apps, right? So they completely were not seeing a practitioner. When they worked with a practitioner who was actually doing the proper workup, they had better outcomes in terms of their weight loss. And so you want to, and that's where the practitioner includes multifaceted pieces of information, not just the labs. Now, but the labs allow you to be more precise. So for, let's say, okay, you're mentioning weight loss. Why has somebody gained weight? That's going to vary based on person to person. You could have one person that's been extremely stressed and they're eating a ton and that's causing them to gain weight. You could have another person where their thyroid is off and it's not necessarily that they're under stress, it's that their thyroid hormone is insufficient. So and you might not otherwise detect that. I mean, with a physical exam, you could assess the thyroid or you could run labs that will assess the thyroid. So this is where you have to have a multifaceted approach. I would say, you know, one thing I probably should have included in this webinar is I am a fan of a multi-disciplinary approach in terms of practitioners. And so if you feel like you want to refer them to somebody who is a nutrigenetics expert or who is an endocrinologist or who is, you know, bariatric expert, like refer the patient to the person you feel like will help them address that facet of health that you are suspecting that is not being sufficiently addressed based on the information you have available. So that's going to be, so that context is going to vary based on the practitioner. Because some people are just health coaches, some people are just nutritionists, some people are board certified physicians, right? They have the whole gamut. So it depends on what they have available, the tools they have. But ultimately, using the information that, you know, especially the omics information, it allows you to be more precise in figuring out nutrition deficiencies, figuring out, so within nutrition, these nutrients are working in metabolite pathways. And so how are the pathways functioning? By investigating that, then you could have a better idea of what pathways to target. And then knowing the pathways, you know, you could see what metabolites can shift those metabolic pathways. So for example, let's say fatty acid metabolism, you could look at different fatty acids in omega-3 metabolism. Those enzymes that promote omega-3 metabolism require cofactors. And so you could look at those cofactors, or you could extrapolate based on where certain metabolites are that some cofactors may be insufficient or in excess in certain circumstances. So, you know, it's, I know you're looking for a more simplistic answer, but it's unfortunately, it's complex, but I would say this is where it should not fall solely on you, not you in particular, but it should not just solely on one person. I'm a fan of a health team. And actually pretty much most of my patients that I have, they have multiple doctors on board that are addressing multiple areas of their health.
And I would want to fill into, and Dr. Holt, I do see your question there. We'll get right to you in just a second. But yeah, I think, you know, what you have to answer, because you can go out into the market and you can find somebody that is going to tell you, hey, the patient should eat this because of why. You know, so for example, I could make up a questionnaire and I could say, hey, if you fill out my questionnaire, I'm going to tell you this diet is going to be best for you, or this diet is going to be best for you, right? I'm taking data and I'm giving you an output. I think the reality of it is that it's more complex for the more complex cases. And that's kind of what Greg is getting at here. So what I would do to make this more practical, we're not here to tell you how to run your clinic. You know, our goal is just to see like, hey, does metabolomics make sense for your clinic? It sounds like maybe not at this point. I'm not 100% sure. You could talk with Greg more in the one-on-one. But I think you have to look at your avatar, like who are the type of people coming into my clinic? How much money do they have to spend? What are their objectives, etc. And then from that, you can identify the resource that you have available. So like, could my population afford nutrigenomics, this, that, and the other? Because then you can create your process around, we run these labs. If you find though, that your patient base typically is like, hey, they got $100-200 to spend, then you know what you're working with, right? Now you know kind of like your resource that you can go to, and maybe you can find something that's not the ideal, but it's good enough for $100 or $200. And I'm not sure exactly what that would be. I will say too that most of the physicians, providers, clinics that work with us typically are pretty experienced in labs, the ones that adopt it and use it quite a lot. So I don't want to say don't start with us, but really the ones that are successful with it generally have experience in labs. Would you concur with that, Greg?
Yeah, I mean, let me put it this way. There's a reason we initially, I mean, Mike, correct me if I'm wrong, but we're initially opening up metabolomics testing to just lay people. And we have since changed that over the last several months to focusing specifically on practitioners, because it's a lot of information. Covering metabolomics is a lot of information. Covering things that involve dietary, lifestyle, supplement changes gets more complex. And so that's where it comes to the practitioner. And the options currently available, like when I see any entities where it's like, input your information, your labs or your genome, and we'll tell you exactly what supplement to do or what diet, it just doesn't work that well. The studies show it doesn't work that well, and in practice it doesn't work well. And I'll see patients who did that and they're like, well, this website had recommended this to me. It's like, this is the role of working with a practitioner, a health coach, a consult, whoever, is that they look at multiple levels of information and they're able to make an accurate assessment. And there's many tools in a tool chest. Metabolomics is a powerful tool. It's an extremely powerful tool. It's probably the most powerful tool within the omics field. And so, but it's going to vary. It's going to vary person to person. Everyone has their own clinical experience, their own things that they've seen and observed. And also, this is where forums like this, where we could talk about these things and what people are seeing. I had someone last week with in the webinar, I think 2 weeks ago, they had a patient with kidney failure, and we were talking about different types of dietary strategies for that and what the labs had shown. So, even patient cases are not cut and dry. So it gets a little more complex. With that said, you have to start somewhere. And I think that's what I was trying to impress upon here. Like there should be some baseline level of nutritional assessment before you're running in metabolomics, because what's your baseline, right? Like that information is even collected in the metabolomics when we're inputting it and the AI is assessing their diet. So, if they have certain dietary restrictions, dietary allergies, things of that nature, we're already capturing some of that information. But that information needs to be collected at some point.
Dr. Holt, I saw you had a question. Do you want to chime in?
Hi, Micah. Thank you. And Dr. Gregory, thank you so much for being with us today. I 100% back up what you're saying. This is an application-based value product, because in clinic, in a real life situation, we have to understand not only our markers and our testing, but also how the patient is expressing and demonstrating on a day-to-day basis. And so I had a question about illness, versus wellness. When we have a patient that comes in and they're well, doing this particular type of study will yield very different results than when we have a patient who is clinically diagnosable. For instance, I have a patient right now with active IBS. She has antibody positive bacteria, like 12 of them. Extremely a lab marker of zonulin over 1,000. And so with that person, that's a chronic case. And at what point do we recognize in science that genetics start to change in response to chronic illness? And then at what point does that begin to alter laboratory testing, right? Is that a fair question? Because I'm trying to understand, with all of the laboratory testing that we have and, genomics and all these things that have kind of been on the market for the last 10 years or so, in a clinical application, how do we pick up the, 126 markers off of your test and be able to use it clinically based on what we know about our patient journey, via one that's very sick, and how does the test reflect that illness versus somebody that just comes in that's well, that may just want to maintain wellness? Is that a fair question?
No, I think that's
I know it's a loaded question, but
No. No, I mean, it makes a complete sense because this is where, again, if somebody was not working with a practitioner, let's say like in this circumstance, this person sounds like their gut is an absolute mess. If their zonulin is elevated, that means their gut is overtly leaking, and that's going to cause massive nutritional deficiencies. And so, am I going to run a nutrigenetic test and be like, oh, you are deficient because you have these couple SNPs? It's like, no, my gut is completely porous, not absorbing anything. And so, now, this is where the metabolite testing would come into play because you would see altered metabolite pathways, especially in the gut. You would see altered metabolite pathways probably within other spectrums of health, like inflammation would likely be upregulated in response to gut leakiness. And then by order of priority, if somebody has severe gut inflammation, it's not, well, let's give you more nutrients. I mean, yes, replete the nutrients. I guess depending on how severe, you might want to send them for IV nutrition, but you want to heal the gut first. And so maybe technology will get to a point where it can identify this, but really it comes down to the practitioner. I mean, I don't know. In a way, I feel like it's going to keep me employed by being the practitioner that can identify the order of operations and say, okay, based on what I'm assessing with your symptoms, the gut is the biggest problem we have to resolve here. So this is where there's I don't want to call it common sense, but there's being a practitioner, there's an order of operations that you know to heal, and especially all disease starts in the gut. I'm a big fan of that. So yeah, that is you know, a good area to focus on.
And thank you so much for that. And I totally agree with you. Prioritization is the name of the game for all of us. Otherwise, our patients are just spending money and throwing it out the window if there's no priority-based order of operation on how to work through what's first, next, and last. I think that's a huge missing piece for a lot of doctors. They didn't get that in school, right? We have struggled to figure that out on our own. But my question, just to clarify one more time, Dr. Gregory, is how are the results different when we have someone who is seeking wellness, right, that is not chronically ill versus someone who is chronically ill running that same panel of testing? At what point does the genetic variation start to truly become valuable for us in a clinical sense?
I will put a preference on, well, okay, nutrigenetics, just looking at your genetic predispositions. And again, your genetic code is not going to change. The expression is going to change. You could look at laboratory testing that's not including nutrigenetics as essentially the output of the genome in conjunction with environmental exposures, lifestyle exposures. So for people that are ill, that's where the laboratory values come much more into play. Quite frankly, if I have a very ill patient, I'm putting a lot less stock on the nutrigenetics because I don't know what their output is. Let's say for MTHFR, let's say, okay, you have a genetic predisposition for MTHFR. I'm measuring homocysteine. I'm looking at methylation metabolites because let's say if somebody, they don't have the MTHFR variant or they don't have variations in their MTHFR pathway, but they're B vitamin deficient and they have excess oxidative stress and inflammation. That's going to mess up the methylation cycle. And so if I just looked at MTHFR, I would say your methylation cycle is fine. You should be totally good. But if you look at the metabolites, you see all the metabolites are off and it's like, okay, actually, based on these metabolite shifts, you are predisposed toward anxiety, depression, inflammation, and we're seeing these shifts in the metabolite pathway. So I would say if for someone that is already completely healthy and they're like, I want to optimize my diet and know what the best like dietary strategies I could take, yes, I would lean more heavily with nutrigenetics. But for ill patients, especially chronically ill patients, I'm putting more stock into the metabolite testing and other laboratory data.
Fair enough. And just one last question. I'm so sorry to take all the time.
No, it's okay.
But one last one. And so just to for my own clarity, when the lab panel is complete, is there a representative on staff who can help walk us through the understanding and teach us how to utilize this in clinic? And second question is, are there protocols that you have attached to some of these outcomes that you already have, researched and looked at, from across a spectrum of demographic?
Yes, absolutely. So if you do the metabolite testing through Therium, we do offer a consult with practitioners who go over those results, and we'll not just go over the results, but we'll go over your patient case. So what is the thing that they're trying to solve, whether they're healthy or have other chronic issues, and then we'll apply it. And then, so we use something called a digital twinning protocol. So basically, from the metabolite pathways we look at, we calculate health scores. And so what we'll do is we have a database of over 1,300 interventions, various different dietary things, supplement things, lifestyle things. And so we'll create digital copies of that individual based on their biological inputs, their metabolite information, and their health history. And then we'll expose that digital copy to these interventions, and we'll see how the health score shifts. And so we'll create a personalized protocol based on what's going to have the most favorable impact on the metabolome. So we do have a process for that. That is pretty
That's
comprehensive.
super valuable. And thank you very much for your time.
Yeah.
Absolutely.
Yeah, and I want to clarify on that too. You know, if you're running like IVs and that kind of thing, we are working like it's really, I find it to be a very valuable piece, right, of our company is that we can produce a protocol. So from the concept standpoint, it's really important for us to keep progressing with that in the sense that we're adding interventions, even interventions that don't get recommended often, because I think the omission of things is just as important as putting something in. But that being said, you know, right now, I think it's going to give you kind of the base construct that's really the foundational construct that's really important to work with the patient on in terms of their supplemental nutrition, what they need to do, some of the exercise, some of the diet stuff. But, you know, it's not going to include like your more, let's say, invasive interventions that you're doing at the clinic at this point. And there's a couple of reasons for that. One is that we need a lot more people on staff that are just dedicated to feeding, you know, this thing with data and understanding. But number two, you know, you start to run into regulatory constraints of what you can and cannot recommend. But my ideal world, and you know, the whole reason I got into this industry was because my brother had cancer, right? And so it would have been really helpful at the time that that happened to understand what should he do and also what should he not do. And so I imagine a world with this where it's making really, really insightful recommendations, even into things like chemotherapy and whatnot. And, you know, I don't know how long it'll take to get there, but just wanted to add that. So we are 20 minutes past. We are going to have to jump off this meeting, but I really appreciate everybody joining. Again, if you want to talk with Greg one-on-one and see if metabolomics makes sense for your clinic, just go ahead and I'm sorry, I'm pimping you out so much, Greg. Go ahead and click that link there, therio.me/train, and you can schedule a time with him. And then you asked about what does training look like? Well, once you get we find that like having actual real results, that could be you, your spouse, your patient, whatever. Once you get your initial test, you're going to be actually going through a hands-on training with Greg through Zoom. And he's going to be walking you through this patient's case in particular. And you're welcome to do that as much as you need until you feel like have satiated your understanding of the test. So that is available to practitioners who buy through us. And we're building an onboarding program or course, so to speak, as well, but we'll always have that hands-on live interaction you can do as well. So thank you guys.